The first distinguished speaker of the day is Dr. K. Thangaraj. Dr. Thangaraj is the current director of the Center for DNA Fingerprinting and Diagnostics in Hyderabad. He is a fellow of all Indian National Science Academies and is also a recipient of several prestigious awards and fellowships. Dr. Thangaraj is well known for his research on the origin of the Indian population. We invite Dr. Thangaraj to deliver a talk on the topic, Host Genetics and Infectious Diseases, COVID-19 Perspectives. We welcome you, sir.
Thank you very much, and good evening to all of you. So I must thank Dr. Moinak Banerjee for organizing this important conference. I also thank Dr. Chandrabas for supporting him. And we had an excellent talk by Professor Majumdar about the virus and how the mutation which took place and several associated stories behind that. So now let's look at how the host genome, that is our own genome, is responsible for infectious diseases. I hope this slide is visible in full screen. Could anybody tell?
Yes,
whether
yes.
Okay, good.
Yes.
Okay, great. As I hope that all of you, most of you are the genetic students and also those who are working on genetics. As a geneticist, we all believe that so everything is involved. Genetics is involved in everything, right from the behavior to infectious disease, because these two are two extreme conditions that we all know that there is any mutation in the coding region or even in the non-coding region now are associated with genetic diseases. But of late, we started understanding that the infection, the mutations or variations exist in the genome is also responsible for other things. Like the chief guest address, he mentioned that the genomic variation is responsible for therapeutic response or adverse response, or even the dose is determined by the mutations in the genes which are metabolized in the drug. When we talk about genome, we all know that we have about 3 billion base pair, there are 46 chromosomes, it's common for everyone. But what makes the difference? The difference is because of the variations that exist in our genome. And if you take two unrelated individuals, you'll find it's about 99.9% similarity. However, that 0.4% makes about 4 million difference in our genome. So that means that these genomes, these variation genomes are responsible for the disease, responsible to drug, and makes individual either susceptible or resistant to infectious diseases. When we talk about infectious disease, I just want to give you examples of some of the disease which have worked earlier. There are 2 parasite disease, malaria and leishmaniasis. I will not talk too much about all this disease, but towards the end, I will talk about COVID. And the rheumatic heart disease is caused by bacteria. Of course, COVID, all of you are aware that's caused by the virus. If you take malaria, there are few studies, even our own studies, we found that there are several genes which are responsible, or the mutation those genes are responsible for making individuals either susceptible or resistant against malaria. Of course, we also know that because of the constant exposure to malarial parasite, certain population have gained some mutation which gives them resistance against malaria. So in this slide, what I'm showing is one of the study where we looked at the Mannose binding lectin gene variations. So where in 3 different categories, one is the severe malaria, mild malaria, and asymptomatic. And this gene has been well studied, and there are mutations which are identified, helped in making the haplotype. there are haplotypes which are associated with high MBL level, that's a serum MBL level, which gives protection against the infectious disease, whereas the low haplotype, which secrete the low amount of serum MBL, gives susceptible against this disease. Where you can clearly see that the bar diagram shows that the low MBL haplotype is high among severe individual, moderate here and mild, and asymptomatic is very low. That means if the haplotype is very high, that means these people are susceptible. On the other hand, you can see high MBL haplotypes are low here and very high among the asymptomatic individuals. That clearly shows that the genomic variation makes individual either susceptible or resistant against infectious disease, particularly in this case, malaria. So there are several other genes which we have studied, like IF and beta-1, non-coding RNA, and even the blood group O is associated with malaria. I'll take you to another parasite disease, leishmaniasis, caused by the protozoan parasite, which was spread by sandfly, and this is one of the tropical diseases. Again, we looked at several genes, MBL, DLL, interleukin genes, and many more. Again, we found that there's a clear-cut evidence suggesting that the genomic variations are responsible for making certain group of individuals more susceptible for leishmaniasis. So now I'll take you to the rheumatic heart disease. Mostly, we all know that the heart disease are caused by genes that are in the either in the sarcomeric genes, particularly causes the cardiomyopathies. But the bacteria, that streptococcus, which causes the infection, initially the throat infection, then that infection leads to rheumatic fever, and from there manifest into 5 different diseases. Among that, the major cause is the rheumatic heart disease. Up to 45% of the people who get rheumatic fever get the rheumatic heart disease. Then some gets polyarthritis, some gets chorea, some erythema marginatum, some subcutaneous nodules, as you see here. But interestingly, none of individuals have more than one disease. That shows that the genomic variation actually responsible for what kind of disease the individual get after the rheumatic fever. So we thought of looking at the genomic variations present in the rheumatic heart disease. And here you see that the mitral valve is the one which actually affects, because the M protein, which is there in streptococcus, is mimicked by the protein which is there, particularly the polypeptide in the mitral valve. Therefore, it targets there, and there's a prolapse of the mitral valve and leading to regurgitation. Therefore, this is a normal valve where the blood flows into the atrium and in the arteries. But whereas here, because of the prolapse of mitral valve, there's a backflow of the blood, and ultimately it leads to heart failure, and somebody has to go for the mechanical valve. And here, look at the streptococcus pyogen infection, and these are the genes which comes into the first line of defense, like MBL, which we have seen earlier with other diseases, Tricolin, TLR, and these are the adaptive immunity gene. Of course, we studied all of these in the patients with the rheumatic heart disease, and I'll just give you a few examples. In Mannose binding lectin gene, again, we looked at the haplotype, which are of high MBL sector and low MBL sector haplotype, and we can clearly see that there's a difference between the control and the rheumatic heart disease, where you see 28% in high MBL sector haplotype and 18% in the patient. And this, if you look at the p-value, it's very significant. Same way, it's other way around in the low MBL secreted haplotype. There are several novel mutations we found, and one interesting mutation is there in polyadenylation site, where this was known earlier, published in nucleic acid research few years back, where they have studied by changing every nucleotide in the polyadenylation site and found the impact of that. Particularly where we found the mutation in this particular site, it reduced the polyadenylation by 95% approximately, and you can see the impact of it. And this is just to compare the control and RHD MBL serum level. You can see significant difference between these two groups, and particularly when we looked at the mutation in the polyadenylation site, and although number is small, but you can see so much of difference, and it's highly significant, again suggesting that the mutation in the MBL gene is very important for all kind of diseases, parasite, and also in bacterial disease. And this is another one which we studied, ficolin, very recently published online, where you can again see this significant difference of ficolin serum level between the case and control. And these are different haplotypes, which again gives a significant difference between case and control. So now let's look at the COVID, because COVID, as Professor Pathamajinder also mentioned, that not much of the host genetic response has been seen because of couple of reasons. Most of the disease where we see is a small proportion of the population has the disease, therefore it's easy to look for the genetic variations. And second is that because of that, where we see that across the globe, you have large number of people are infected. And the second one, because of that, the cost involvement in analyzing the genome of large number of individuals is very, very difficult. Therefore, not much study, although there were large number of studies has been done, but not many when compared to the other aspects of COVID genome, COVID study, including COVID genome sequencing and so on. So I just want to give you a couple of studies. One study which was published some time ago, about a year ago, by Fabo, is one of the well-known scientists who have extensive study on Neanderthal and looking at the ancient DNA in the context of human evolution. He looked at a large number of patients, about 3,199 patients severely affected with COVID hospitalized. He also compared about 9 lakhs controls, both from UK, and he found there's a 50 kb core Neanderthal haplotype was significantly present among the South Asians who have been hospitalized, about 50% against 16% in the European. Therefore, he suggested that maybe the 50 kb region which we inherited from Neanderthal may be responsible for causing the disease or increasing the susceptibility for COVID-19. And what is the frequency? And he has seen, and you can see in the map, in South Asia, particularly India and Bangladesh is even more higher, that's a 63%. of this particular proportion of DNA segment which we inherited from Neanderthal. So to give you some idea about Neanderthals in the context of human evolution, Neanderthal is very close in terms of close to human in terms of evolution. So what is interesting is that the Neanderthal had slightly larger brain compared to human. If you look at the evolution, and in the early species, brain size was as similar as chimpanzees and increased to 35%, 50%, and so on. And we don't know whether it's really intelligent or not, but this species is vanished. There's an interesting theory told by Paul Mullers from Cambridge that inherent violent behavior of modern human probably responsible for vanishing of the species. That's in context of human evolution and vanishing of this particular species. So some time ago it was known that the Neanderthal have interbred with the modern human, but not with the Yoruban and San, that's in Africans, but the remaining people. And few years ago, there's a large number of studies, 300 genome worldwide from 142 population. We are also part of it. As you see here, these are the places where the genome was sequenced, including large number of samples from India. And you can clearly see that the African doesn't have Neanderthal genome, whereas rest of the populations have Neanderthal genome. Particularly the East Asians have much more, that is up to 3% of Neanderthal genome. And when we looked at, we thought that let's look at, because Savanthapabo and his colleagues have highlighted that the 50 kb region which we inherited from Neanderthal might be the susceptible factor for COVID. We try to look at that, and in fact, this shows that, so this is the AC2 variation which people have already reported, which shows slightly significant p-value, whereas the RS number, which is a part of that 50 kb region, is not showing actually any significant for this. So suggesting that although the people who are hospitalized in Europe, who basically have gone from South Asians, have high frequency, but could be several other factors responsible for that. The way they have lived, the hygiene, and many more things. Therefore, we cannot actually look at that the South Asians are more prone for COVID infection because of inheritance of the 50 kb region. And I also wanted to highlight about another study, basically from Brazil. There are large number of indigenous populations, and they are the one who are highly affected. There are 3 studies which we looked at, a couple of them from Lancet and one from Frontiers in Psychiatry, where they have shown that incidence index of the COVID-19, and this is a mortality index where you can clearly see that most of the indigenous people, and also there's a report showing that they are underreported. So that means that they are more susceptible, and the reason I'll give you sometime later. We also thought of looking at different Indian population groups, including the Andamanese, where we have the tribe which are in isolation, and also I'll give you the background of that in the coming slide. They are in isolation, small population, and there's no connection so far with the modern human, and they're not exposed to any modern medicine, probably not exposed to some of the pathogens. Therefore, they are more susceptible. In terms of their genetics, when we looked at large number of linguistically isolated groups, including Onge, so we have Nihalis, and there are groups in Nepal, like Kusundats. So they are all linguistically isolated groups. When we looked at the mean length of the DNA segment, that's in KB, 150 KB region, and also the runs of homozygosity. So if you look at the Onge, and there are people particularly from South India where they practice the endogamy, marrying within the group, and small population like Onge, Nicobaris, they are the one who are having such a high runs of homozygosity and much longer DNA segment. So that we found, and you can see that Java and Onge from Andaman and Nicobar Islands are having a very high significant level of this runs of homozygosity. And this paper just got accepted in Genes and Immunity. I was talking about the reason why they are more susceptible. I'm just going to show you one of our earlier study on malaria again. We looked at interleukin-4 gene and considered the haplotype, and these three mutations, minus 590T, minus 334T, and intron, that's a VNTR repeat R2. So these are giving much more resistant against malaria. And if you look at the data, asymptomatic, mild malaria, and severe malaria, there are three which gives more susceptible, R2 gives more resistant, irrespective of whether they are mild malaria or severe malaria, they have high proportion of R3. On the other hand, in the context of different populations, we looked at the caste population, nomadic people, tribes, and primitive tribes, something like the Onge and many other tribes which are in the isolation. So they have the TTR2 that gives more susceptible against infection, have high frequency of that. So that tells that because they are in isolation, probably they are not exposed to the parasite to develop some kind of new mutation which gives the resistance against some of the disease. So this gives the idea that people who are in remote area, who are not exposed earlier to this kind of diseases, are more prone for such a disease. And again, to give you some idea about Andamanese, who has a high runs of homozygosity. So some of you who are not aware of Andamanese, there are 4 tribal groups who typically look like Africans, as you've seen here. Jarawa, Great Andamanese, Sentinelese, and Onge. They are very small in number. And several years ago, so we have studied their genome by collecting sample from Onge, Great Andamanese, and Jarawas. And we found that they are the first modern human who migrated out of Africa, taking southern coastal route, and they have reached Andaman and Nicobar Islands probably around 65,000 years ago, and they are still in isolation. The Sentinelese are still in isolation, nobody can go near them. They are in a very pure environment, they are not exposed to any kind of the pathogen or pollution. Therefore, they are much more susceptible against any kind of infectious disease. So to conclude my talk, not in broadly with respect to COVID, that the 50 kb region which we inherited from Neanderthal have no significant association with the disease. The real-time data in South Asia also do not support the high susceptibility of South Asian population compared to European. Longer runs of homozygosity population may have higher risk for COVID-19 or any other infectious disease. Andaman Islanders have the highest number as well as longest voyage segment. Therefore, we suggest a high priority protection and utmost care should be given to such kind of people who are the treasure of modern human evolution. So with that, I would like to thank all my colleagues within CCMB. There are many more, and the collaborators. And Janeshwar contributed a lot in the last part of the study, and many more. The list is goes, and these are the funding agencies. And with that, I end my talk, thanking all of you for listening. Thank you very much.